In this video, we're going to do an overview of anatomy and physiology of the small intestine. So the first point to make, which I think should be obvious, is if you watched our last video, the small intestine gets its contents immediately from the stomach. This picture isn't showing the stomach, but it does show these three different portions of the duodenum, the jejunum, and the ileum. Those are the three different segments of small intestinal tissue. And then after you're done with the ileum, the next step is going to be moving into large intestine that we'll get to in a little bit. Right. If you zoom in on this tissue, the interesting stuff is really happening on a microscopic level. Right. These structure that you see here are called villi, and villi are basically just big folds in the tissue that they look like fingers, like projections coming out of the tissue. They're there entirely to increase surface area. And that's kind of the name of the game with the small intestine is more surface area means more membrane, means more transporters can be put in the epithelium, and so we can absorb more nutrients. Right? This is what it looks like under sort of a 3D view from an electron microscope. Each of these things is one villus, and you can't see individual cells on this, but all of these guys up here, these little rectangular shaped cells, those are the cells that you would see if you could dig into that villus, and the food that's surrounding them needs to have its molecules and nutrients moved across that epithelial cell because deep inside this villus, again, it's not really visible here, you're going to have blood vessels that need to take those nutrients and transport them out to the body. Okay, so this illustration gives you a better idea of what's going on. In a zoomed out view, right, these villi that you're seeing up here, right, are the same thing as these villi that we're seeing down here. And the villi, if you zoom out, you get a view of these circular folds, which are just further curves in the tissue, right? And this zoomed in view gives you an even better view. This is one villus. This is another one. That's another one. The goal in all cases is, again, surface area. So folds in the tissue, villi under a microscopic view, and then if you zoom in really, really close, like right on the surface of one of these villi, you will see microvilli. Right? This is a single cell down here, an intestinal epithelial cell, and these microvilli are just the membrane weaving in and out over and over again to massively increase surface area. Right? You can imagine if the surface of the cell was just flat like this, as opposed to, right, this microvilli pattern, we can multiply the surface area many, many times over to the point where an entire small intestine in one person can have about 800 or 900 square feet of cell membrane because of all these surface area enhancers. Right? And the goal here is, right, this would be the lumen of the small intestine. The idea is we want to get nutrients from that lumen, whether it's glucose or other carbohydrates, amino acids, vitamins, minerals. We need to transport those nutrients across the epithelial layer. Some of those nutrients, like fats, are going to wind up in these lacteals, which are basically capillaries of the lymphatic system. Most of them are going to wind up in the arteries and arterioles, right, that are going back to blood supply. And a lot of that, all of that in the GI tract runs through the portal vein, which is something that you'll see once we get to the liver, right? So the next thing to point out is why exactly do we need all this membrane surface area? It really boils down to needing transporters, right? These epithelial cells are supposed to be selective transporters so that they only let nutrients that we want come into the cell. And they don't allow toxins and waste products and bacteria and things like that to get through. And so since everything has to actually go through the membrane, remember, most molecules don't diffuse directly through cell membranes, and so you need some sort of transporter inserted in the membrane to make it happen. Now, pretty much every nutrient that a person can absorb from their food needs its own transport system. The purpose of this class isn't to go into that much detail. And so I'm going to use one particular system as an example. Right? This is showing you the absorption of glucose and sodium. They happen to use a shared transporter. It'll make sense why in a minute. So in this graphic, this is the inside of one intestinal epithelial cell. and this would be the lumen of the small intestine. So this is where you would find whatever food that person just ate, and then underneath that epithelial cell, on the basal side, you have blood. Your whole goal is to get your nutrients, move them across into the bloodstream as efficiently as possible. Now, the problem is, it's not enough to just let these molecules move by diffusion. And the reason is, diffusion will only go until you reach equilibrium. So, as an example, if you let these glucose molecules move simply by diffusion, they would keep moving this way until the concentration in the lumen and the concentration in the blood were equal. We don't want that, because that means we're leaving a bunch of glucose in the small intestine and not absorbing it. And so what ends up happening is we move glucose and sodium together through this sodium-glucose cotransporter, which just means it's a pump, really an indirect active transport pump, right? So this thing does not use ATP itself directly, but this sodium-potassium pump over here does, right? So this is an ATPase, which means it's a primary active transporter, right? This pump is moving sodium out of the cell into the lumen of the blood. The end result of that is you're going to have very low sodium levels inside the cell. That sets up a gradient so that sodium levels are always moving from high to low in this direction, right? What that means is sodium always wants to go into the cell, and it can effectively drag glucose along with it, right? The end result of that is, even though this isn't a direct active transporter for glucose, you can pull glucose against its concentration gradient and move it in a direction that it wouldn't normally want to go, right? Eventually from a low to a high concentration. And so all the energy in the system is being put into the sodium-potassium pump. We're just using the pull of sodium to drag glucose along for the ride. And then you have a single glucose transporter on this side that acts like an overflow valve. So as glucose gets pulled into the cell, it will overflow through this transporter into the blood, and then you can use insulin to store that glucose as glycogen or as fat and save it for later. Okay, so remember, every nutrient that you can absorb needs a system somewhat similar to this. They work by different mechanisms, right? Different transporters, active transport pumps, and the reason you have all those microvilli is so that you can put more of those transporters in the small intestine and try to increase the rate of nutrient absorption. Okay. All right, a few other things with the small intestine that we should tackle. One is, remember, one of the purposes of hydrochloric acid in the stomach is to kill pathogens, right? Bacteria, viruses, but it's not perfect. Some bacteria, just by chance, are going to survive. And so in the chyme coming out of the stomach, there is not very much in the way of bacterial growth, but as food gets neutralized and moved through the small intestine, those bacteria are going to regrow. And so one thing that we see in the ileum, near the end of the small intestine, are these structures called Peyer's patches, which are a lot like lymphatic tissue. They're just packed full of white blood cells. And the idea is, if you have bacteria that happen to wiggle their way through your mucosa, this is like a protective barrier for the immune system to be ready to detect any of those bacteria or other pathogens that happen to make their way through, and hopefully, kill them off before they get into the bloodstream and cause something horrible like sepsis. Right. The second point to make with this is everyone has bacteria in their GI tract, and having none is impossible, and it wouldn't even be healthy anyway. So there have been research projects recently, like this Human Microbiome Project, that are trying to ask the question, what kinds of bacteria are good for digestion and good for people, and which ones are pathogenic. And the idea is, if we can encourage the growth of bacteria that are beneficial, then hopefully they can outcompete ones that are harmful, right? Things like E. coli or C. diff or salmonella that you obviously don't want growing in your GI tract. So a few interesting bits of research. One is, the title of this paper kind of explains itself, right? Diet rapidly and reproducibly alters the human gut microbiome. All that's saying is the food that you eat very, very quickly and dramatically controls what bacteria grow inside of you. And it makes sense because the leftover bits of food that you eat, that is the food for your bacteria. And so all they did in the study is they compared people that primarily ate an animal-based diet, and they compared it to people that primarily ate a plant-based diet. And the point of the study wasn't to ask which one's healthier compared to the other. All they were getting at is when you did switch back and forth from mostly plants to mostly food, from animal products, you dramatically altered the kinds of bacteria that grow in the GI tract. Okay? And so when people take something like a probiotic, just remember, if you don't feed the bacteria that you're putting in your GI tract the kind of nutrients that they need to thrive on, they're not going to grow. And so taking a probiotic and then eating a bunch of junk food that doesn't have any fiber or nutrients with it probably isn't going to get the job done. All right. The second interesting study that I decided to throw in, this experiment was done in mice, but they did a cool thing. They took microbes, gut bacteria, from obese mice, and they transferred those bacteria into lean mice. And what's crazy is, without changing the food that the mice were eating, just moving those bacteria to the lean mice induced obesity, meaning it made them gain a bunch of weight. And the wild thing is, it happened because, one, it increased the amount of energy that they could extract from their food, so they were getting more calories, and it increased their appetite. That's kind of crazy because it means that something about these gut microbes was affecting the brains of those mice, because appetite is controlled in the brain. And so apparently the kind of bacteria that grow in your GI tract can influence how hungry a person is, right, by altering their brain chemistry. Okay. All right, just to finish this video off, since it's a pretty short section, a few quick notes on large intestine. The large intestine is getting its contents from the ileum, right, so the end of the small intestine, through this ileocecal valve. And from there, right, we're just going up the ascending colon, first part of the large intestine, across the transverse, and down the descending, and finally to the sigmoid colon at the end. And there's not really an awful lot happening here, so the anatomy is fairly straightforward. The histology, you see a structure or set of structures that look fairly similar to what you saw in the stomach. The difference is almost everything coming out of these intestinal glands, it's just mucus, right? We're not secreting acid at this point. We're not secreting digestive enzymes. So these glands that you see in the large intestine, all of these tiny little cells that look like they're teardrop-shaped, like open compartments, those are goblet cells, right? Those are the mucus-secreting cells of the intestinal gland. And they're just there to keep this large intestine lubricated, keep contents moving through, and they will absorb water and some electrolytes and things like that. But anything that has calories in it, like glucose or fats or amino acids, should hopefully be completely absorbed at this point. Okay. All right, hope that was a helpful introduction into intestinal physiology. And in the next video, we're going to move on to accessory organs like the liver and pancreas. So follow along in the playlist. It'll make more sense if you go in order.